Hello, students. Welcome back to our science session. Today, we are diving deep into what I like to call the botanical blueprint. Have you ever looked at a flower and thought it was just a pretty object? Well, think again. From an evolutionary standpoint, a flower is a highly precise, beautifully engineered machine designed for one ultimate purpose: sexual reproduction. Let's break down how this mechanical masterpiece works. Before we look at the machinery, let's understand the why. Biology is fundamentally the story of life and survival against extinction. While individual organisms eventually die, a species persists through time because of reproduction. Sexual reproduction is a brilliant strategy because it creates new genetic variations, giving species a survival advantage in an ever-changing world. And when we talk about understanding plant reproduction, we must honor a true pioneer, Professor Panchanan Maheshwari. Working at the University of Delhi, this world-renowned Indian botanist revolutionized the field by pioneering test tube fertilization and introvariant pollination. He showed us how we could interact with the plant's reproductive blueprint directly in the lab. Now, let's look at the factory floor. A flower houses two distinct, highly specialized manufacturing centers that develop sequentially. On the male side, we have the androecium, made up of stamens. Each stamen has a terminal head called the anther held up by a stalk called the filament. On the female side, we have the gynoecium, or the pistil system. It features a top landing pad called the stigma, a long transit tube called the style, and a basal chamber known as the ovary. Together, these parallel systems work to manufacture the ultimate end products of plant reproduction: fruits and seeds. Let's zoom in on the male manufacturing center, the anther. Structurally, a typical angiosperm anther follows a dithecous design, meaning it is bilobed and contains four distinct chambers called microsporangia. Think of these as highly secure storage vaults where pollen production takes place. If we take an even closer look at the microsporangium wall, we see a brilliant layer-by-layer design. The outer shield consists of three layers: the epidermis, endothecium, and middle layers. Their job: protection and helping the anther burst open, a process called dehiscence, to release mature pollen. Deep inside sits the inner core, the tapetum. Its cells have dense cytoplasm and multiple nuclei because they have a massive job, providing active nourishment to the developing pollen. Finally, right at the center lies the payload, the sporogenous tissue, packed tightly with cells waiting to undergo meiotic division. This brings us to microsporogenesis, the literal pipeline of pollen production. Starts at step 1, where every single cell in that sporogenous tissue acts as a potential pollen mother cell, holding a diploid 2N set of chromosomes. In step 2, meiotic division occurs, having the genetic material. By step 3, we get a microspore tetrad, a cluster of 4 haploid N cells. Finally, in step 4, as the anther dehydrates, the tetrad dissociates, freeing independent, mature pollen grains ready for active transport. Now, look at this mature pollen grain. It is essentially an armored capsule. The outer wall, or exon, is made of sporopollenin, the most resistant organic material known to science. It can withstand high temperatures, strong acids, and cannot be degraded by any known enzyme. But how does the plant get inside this armor? Through germ pores, deliberate gaps where sporopollenin is absent, acting as an escape hatch for the pollen tube. Beneath the exon is the intine, a smooth, continuous inner membrane made of simple cellulose and pectin. Inside that armored shell is a smart, two-celled transport system. First, there's the massive vegetative cell, which holds abundant food reserves and an irregular nucleus. It acts as the internal life support system during transit. Floating right inside its cytoplasm is the smaller, spindle-shaped generative cell, which carries the actual genetic payload. Fun data point for your exams. In about 60% of flowering plants, pollen is shed at this exact two-celled stage. In the remaining 40%, the generative cell divides one more time before shedding, making it a three-celled system. Pollen presents a fascinating biological paradox. On one hand, it can be a severe environmental hazard. Microscopic airborne grains can trigger intense allergies, leading to chronic respiratory issues like asthma. In India, an invasive weed called parthenium, or carrot grass, is a notorious culprit behind widespread pollen allergies. On the other hand, pollen is a nutritional goldmine. It's packed with nutrients and is processed commercially into tablets and syrups to boost the performance of athletes and racehorses. And if we need to store these precious cells for years, we use cryopreservation, freezing them in liquid nitrogen at a freezing -196°C in specialized pollen banks. Let's shift our focus to the female center, the gynecium. As we mentioned, it consists of the landing platform or stigma, the elongated transit tube or style, and the basal bulged ovary containing chambers called locules. But plants like to mix things up. If a flower has multiple pistils fused tightly together into a single unit, we call it syncarpus, like you see in papaver or the poppy plant. If the multiple pistils are completely free and unfused, we call it apocarpus, which is beautifully illustrated in Mechelia. Nestled safely inside the ovary is the megasporangium, commonly known as the ovule. Think of it as a highly shielded vault. It attaches to the placenta by a stalk called the funicle, and the point where the stalk joins the main body of the ovule is the hilum. The vault is protected by double-layered outer envelopes called integuments, which safely enclose a mass of nutrient-rich cells called the nucellus. At one end, there is a tiny, deliberate opening called the micropyle. This is the exact entry gate for the incoming pollen tube. Directly opposite the micropyle is the chalaza, representing the basal structural pole of the ovule. So, how do we get the female gamete inside this vault? Through megasporogenesis. It begins when a single, large megaspore mother cell differentiates at the micropylar end of the nucellus. This cell undergoes meiosis to form a linear tetrad of 4 haploid megaspores. But look at this incredible twist of ruthless efficiency. 3 of these megaspores deliberately degenerate and die. Only one functional megaspore survives to build the entire embryo sac. We call this exclusive pathway monosporic development. The lone surviving megaspore now goes through an intense construction phase requiring 3 successive rounds of mitosis. This gives us the 2-nucleate, 4-nucleate, and finally, the 8-nucleate stage. Here is your key conceptual insight. These initial divisions are strictly free nuclear. That means the nuclei replicate rapidly in the empty space, floating freely without any immediate cell walls being laid down. Wall formation only happens after the nuclei are in position. The final result is an architectural marvel, a mature embryo sac that is 7-celled but holds 8 nuclei. Let's count them together. At the chalazal end, we have 3 antipodal cells. In the dead center sits a single, massive central cell containing 2 separate polar nuclei floating together. And at the micropylar end, we find the crucial egg apparatus. It contains 2 supporting cells called synergids and 1 vital egg cell. Notice those finger-like projections at the base of the synergids. That is the filiform apparatus, and its sole mechanical job is to guide the incoming pollen tube right to the egg. Now that both systems are mature, we face a major biological dilemma. In flowering plants, both the male and female gametes are entirely non-motile. They cannot walk, swim, or move on their own. The embryo sac is locked deep inside an ovary, while the pollen grain is stranded up on an anther. Bridging this physical gap is the ultimate prerequisite for the survival of the species. The inevitable evolutionary solution, a specialized logistical transport system we call pollination. Depending on where the pollen comes from, nature uses 3 distinct transfer mechanisms. First is autogamy, or true self-pollination, where pollen lands on the stigma of the exact same flower. This requires perfect synchronization in timing and results in 0 genetic diversity. Second is geitonogamy, where pollen moves to a different flower, but on the same plant. While it functionally looks like cross-pollination, because it requires an external agent like wind or an insect, it is genetically identical to autogamy. Third is xenogamy, true cross-pollination, where pollen travels to an entirely different plant. This is the only mechanism that introduces fresh, new genetic variants into the species. To ensure survival, some plants use an incredible evolutionary backup plan called cleistogamy. Take a look at this plant. Above ground, it has normal, open chasmogamous flowers that invite cross-pollination. But look closer near the roots. It also develops hidden, underground cleistogamous flowers that never open. Inside these closed buds, the anther and stigma are in permanent physical contact. Cleistogamous flowers completely sacrifice genetic diversity for a 100% guarantee of setting seeds, ensuring reproduction even in harsh, isolated environments completely devoid of pollinators. Nature leaves nothing to chance. When plants don't use animals, they rely on abiotic agents like wind and water. But because nature can't control the elements, this is a pure game of fluid and aerodynamic probability. To beat the odds, wind-pollinated plants, like corn, feature well-exposed stamens to easily scatter pollen, a massive output of ultralight, non-sticky pollen grains, and large, feathery stigmas acting like nets to trap airborne particles. Water-pollinated plants, like Vallisneria, display a different strategy. The female flowers float on incredibly long stalks right at the water's surface to catch passively drifting male flowers. And to protect their valuable cargo from getting soaked, the pollen grains are shielded by special mucilaginous coverings. Notice how these flowers aren't colorful or full of nectar. That's because wind and water don't care about pretty colors. Energy is strictly saved for mechanics. On the flip side, biotic pollination is a buzzing sensory economy driven by rewards. To attract animals like bees, butterflies, and beetles, plants invest heavily in advertisements. Large, vibrant petals, clustered flowers, and intoxicating fragrances. But animals don't work for free. The flower pays them in physical currency, rich, sugary nectar and nutrient-dense pollen. As the animal brushes against the reproductive parts to gather its food, its sticky body gets heavily dusted with pollen grains, mechanically transferring them to the next flower it visits. It's a perfect business partnership. Sometimes, this partnership becomes so deep that it turns into obligate mutualism, where neither species can survive without the other. A classic example is the relationship between the yucca plant and the yucca moth. The flower offers a highly specific, safe, hidden space, the locule of its ovary, for the moth to deposit its eggs. In return, the moth uses its specialized proboscis to ensure targeted, precise pollination of that specific flower. The moth larvae hatch at the exact moment the plant's seeds begin to develop, feeding on just a few seeds while leaving the rest to ensure the plant's next generation. They are locked in a beautiful, lifelong evolutionary embrace. In summary, from the highly armored, resilient structure of the microscopic pollen grain to the complex, filiform guidance systems of the embryo sac, flowering plants represent millions of years of brilliant evolutionary engineering. They ensure the continuation of life on Earth through the absolute perfect alignment of structural form, precise timing, and ecological partnerships. Next time you see a flower, remember, you are looking at a masterfully designed biological